I'm Mark Williams. I'm professor of physics at Northeastern University in Boston. So my research is generally in the area of studying nucleic acid interactions. So we study a range of different interactions from small molecules that bind to DNA that are anti-cancer drugs or potential anti-cancer drugs to bacterial model replication systems, trying to understand how bacteria replicate, and also HIV replication, so viral replication for viruses that infect human cells. And also we study the packaging of DNA in human cells and also how proteins can access that DNA. So we study a range from the simplest biological systems to what happens in human cells, and work with a lot of collaborators to do single molecule experiments to understand the biophysics and do analogous experiments in cells and try to understand the function of these proteins that we're studying. So optical tweezers, a lot of people say, well, why do you want to apply forces to this? Well, there aren't necessarily forces applied in the biological system, but forces allow you to sort of provide a perturbation to the system to understand how it reacts and to get it to do things that it maybe does only rarely. So an example of this is studying HIV replication, where we look at proteins that are involved in sort of facilitating replication of the RNA in the HIV RNA genome. And you have part of it forms like the RNA hairpin that's very stable, and this protein helps to destabilize that hairpin. And when the protein's not there, the probability of the hairpin opening is very, very small. So you'd have to sit there for days and wait for it to open. But we want to understand the difference between what this protein is doing and what happens when it's not there. And so what you can do is you can actually apply a force and get it to do something that it does only very rarely. And so then you observe that opening of the hairpin, and because you're applying a force and you know what the force is and you know what the distance changes are, you know the energy, and so not only do you get it to do things that it doesn't normally do, but you do it in a way that you can understand how that extrapolates back to zero force and what's the whole energy landscape of the interaction that you're trying to study. And you wouldn't be able to get that information without applying forces and studying it with instruments like this, like optical tweezers. When we study small molecules interacting with DNA, we study one aspect of the binding that might be important as a drug. So some of the small molecules that we study are drugs or are potential drugs. But the idea is that we want to understand how those drugs work, and then once we have an understanding of, this binding mode does this to DNA, then the hope is that you can refine currently existing drugs or design new drugs using rational design that have those characteristics that have been optimized based on information that you obtain in single molecule experiments. So that's one, and we also study HIV replication with the idea being to understand how proteins are involved in HIV replication so that you can target those proteins and their interactions as an anti-HIV drug, for example. Well, I think the general idea of in single molecule experiments, you're able to isolate a single set of events, and when you do that, you can see how heterogeneous the biological world really is. So you observe one at a time, but you observe it many, many times, and you see what you would observe in a bulk experiment or an ensemble experiment is an average of all those, and you wouldn't really be able to see rare events. And sometimes rare events are the things that nucleate the main process you observe that you just might not see, some small fluctuation, opening and closing of a protein or something like that. So the recognition that that's what happens in most biological systems, that there is this distribution, there are, and that you can observe those and then learn both what's happening to a single molecule, but also how that propagates into what's happening to a full biological system.